Okay, students, so today the lecture is about continuous and batch type culture techniques. So, as you have been studying production of antibiotics by the process of fermentation, so this continuous and batch type culture techniques, these are actually the techniques used for the process of fermentation in which different types of products, valuable products are being produced, generated, and then they are purified. So, we will be talking about these fermentation types of techniques which are used for the growth of the microorganisms or growth of the primary metabolites or secondary metabolites. So, moving towards the topic, so for growing cultures, we use normally the fermentation technique, and within the fermentation technique, there are different sub techniques which are used in the process. So the cultures can be grown in three classical operational modes. So there are basically like three types of fermentations which are carried out for the production of valuable products. So first one is batch mode, second one is continuous mode, and the third one is fed batch mode. So for the past 3 decades, there has been tremendous growth in the use of fed batch reactors in fermentation technology. So you can see that in the recent years, the third technique of fermentation, which is called fed batch mode or fed batch technique, so that is being used in the process of fermentation for the production of valuable products. Now, the biotechnology, chemical, and pharmaceutical industries prefer to use the fed batch process due to certain advantages. So, there are like certain um, plus points for using this technique, and when you are using this technique, so as a result you get high yield, pure product, and certain other advantages which we will discuss in the coming slides. So batch and fed batch processes, they are used to handle usually low volume high value products such as fermentation products including amino acids, antibiotics, and chemical compounds. So you can see that in case of the batch mode and fed batch mode techniques of the fermentation process, basically the you're using a low volume. So it means that within the fermenter, the volume is low, and high value products, which means that the product you are getting as a result of fermentation of process are very important. For example, antibiotics, chemical compounds like citric acid, acetic acid, amino acids. You know that amino acids are very important. They are given as a supplement to the people who have deficiency of certain amino acids. So due to the high value of these products, profitability can be improved greatly by optimization and improvements in yield and productivity. So here we are talking about the optimization of the process of fermentation within these sub fermentation techniques. So optimization in yield and productivity. So yield means that the percentage of the product which you are getting, and productivity means that how much product you are producing as a result of this process. So when you are optimizing the conditions like the pH, the temperature, the other factors which are involved or responsible for the growth of the product, so they are being optimized, either they are being decreased or increased as per the uh, productivity details. So now moving towards batch culture. So in a batch operation, all necessary medium components and the inoculum are added at the beginning and not during the period of fermentation. So you have to remember that in case of batch process of fermentation, everything is added at the start of the process. The medium concentrations are not controlled but are allowed to vary as the living cells take them up. So, the medium concentration means that the food which you are giving or you are adding to the fermentation tank, so that is not decreased or increased, but at the start of the process it is being added to the fermentation tank, and then as the process goes on and the microorganisms they are growing, so they are utilizing those ingredients, and as a result the medium concentration is either decreasing or increasing depending upon the type of microorganisms which are growing and the type of the product which is being produced. So the products, be they intra or extracellular, are harvested only at the end of the run. So in case of a batch process, the products which are obtained, either they are antibiotics or chemical compounds, so either they are present inside the microorganisms or they are excreted by the microorganisms outside the cells. So they are only purified, separated out, isolated at the end of the run. So when after 4 to 6 days, when the run has been completed and maximum product has been produced, so then the um, isolation and purification of the desired product is taking place. So in case of batch culture, basic controls for pH, temperature and dissolved oxygen are applied during the course of batch culture. So it means that you are maintaining the pH, you are maintaining the temperature, as well as you are maintaining the supply of oxygen during the whole process of batch culturing. The pH, dissolved oxygen and temperature are normally held constant during the course of batch reactor operation. So it means that the pH is maintained. For example, fermentation process requires a neutral pH or slightly basic pH around about like 8 or 9. So then during the whole process from start till the very end, the pH will be maintained at 8 to 9. So if the pH is decreasing, then uh, specific compounds like sodium hydroxide or any other bases, basic solutions, they will be added to the fermentation tank so that the pH is again maintained at the specified range. Similarly, dissolved oxygen and temperature, they are also maintained. So dissolved oxygen, so the oxygen is added. If the oxygen is consumed by the microorganism, so then the oxygen will be supplied, as well as the temperature. So if the temperature required is, let's say, 40 centigrade and the temperature reaches up to 50 centigrade, then there are like water cooling uh, jackets around the fermentation tank which are responsible for lowering the temperature. So that's how like all these three parameters, they are being maintained constant for the whole process. The only optimization parameters are the initial medium composition. So it means that the food which you are providing to the microorganisms for their production, for their growth, so that food concentration, like how much carbohydrates or fats or proteins you have to provide to the medium, that is being set and optimized at the start of the process. So now moving towards continuous culture. So in case of continuous culture, one or more feed streams containing the necessary nutrients are fed continuously, while the effluent stream containing the cells, products and residues is continuously removed. So it means that as you are adding the media, as you are adding the food for the growth of the microorganism, so along with that, the cells, products and the residues or debris which is produced, that is also being removed. So with the passage of time, step by step, you are adding food, providing food to the microorganisms, and at the same time you are removing the product as well. So in a continuous culture, you are adding the food time by time, as well as you are removing the products and other cell debris time by time. So it means that you are not stopping the addition of the medium, as well as you are not stopping the removal of the product during the course of the process. So as a result of this, a steady state is established by maintaining an equal volumetric flow rate for the feed and effluent stream. So it means that the amount of the food or medium which is added to the fermentation tank is equal to the amount of the product or the cell debris which are being removed. So that's how a steady state is maintained. The culture volume is kept constant and all nutrient concentration remained at constant steady state values. So it means that the whole volume of the fermentation tank remains constant. For example, it is, let's say, 40 liters, and you are adding 10 liters of the food or medium to the fermentation tank, so it becomes 50 liter. And then at the same time you are removing the 10-liter product from the fermentation tank in the form of antibiotic or cell debris or residues. So again, you are left with the 40 liter of the volume of the fermentation tank. So that is how during the course of the continuous culture technique, the volume is maintained at a specific uh, rate. So that's how you are establishing the steady state uh, equilibrium. So continuous batch operations are common in chemical industries. With the exception of single cell protein production, certain beer production and municipal waste treatment processes, continuous cultures have not been adopted widely by industry. So here you can see that like in most of the cases, the continuous culture technique is not used in the industries, like in the pharmaceutical industry or in the chemical industry, but basically only in single cell protein production and in the production of beer, this technique is being used, as well as the waste treatment processes. It is not dominant mode of industrial operation primarily because of the difficulty in maintaining sterility, free of contamination by other organisms, and protection against phage attacks. So it means that when you are in the case of continuous culture, when you are adding the food or media into the fermentation tank, and at the same time you are removing the product from the fermentation tank, so there is a possibility, like big possibility, that the contamination may occur. There will be other microorganisms which will be growing during this process because you are adding the media. So it means that there is a inlet open and outlet is also open, so there is a big chance, a big probability that contamination can occur and the culture is not pure. Phage attacks means that viral attacks, so viruses may enter into the fermentation tank and they may attack the bacteria or microorganisms which are responsible for producing the product. So in that case, the continuous culture is not liked or is not preferred by the industrialists for the production of certain valuable products. Often, steady state operations are found to yield poorer results than dynamic operations. So it means that the yield, the production of the product in case of continuous culture is not maximum, is not like at the highest level in comparison to other batch mode or fed batch mode. So that's why this technique is not like preferred by most of the industries. Right, moving towards fed batch culture. So a fed batch culture is a semi-batch operation in which the nutrients necessary for cell growth and product formation are fed either intermittently or continuously via one or more feed streams during the course of operation. The culture broth is harvested usually only at the end of the operational period, either fully or partially, the remainder serving as the inoculum for the next repeated round. So the difference between continuous culture and the fed batch culture is that in case of fed batch culture, you are adding the media, you are adding the food to the fermentation tank intermittently or continuously. Intermittently means that after specific periods of time, and continuously means that like without any break. But at the same time, the difference between fed batch culture, and the continuous culture is that in case of fed batch culture, you are not removing the product. So the culture broth is harvested. Harvested means that you are taking up the product and then at the end of the operational period. So when the run has been completed and the maximum product has been produced, so then you are removing the product, and either fully or partially, the remainder serving as inoculum for the next repeated run. So the rest which has been left inside the fermentation tank, so that is serving as inoculum. So it is serving as a starting culture for the next repeated run. Next repeated run means that you are again doing the next fed batch culture, in which the remaining media has being utilized. It is being utilized for the next fed batch culture, in which again you are adding the media continuously or intermittently. So basically, the difference between fed batch culture and the continuous culture is that in fed batch culture, you are continuously adding the media intermittently or continuously, but you are not taking out the product at the same time. The product is taken out at the end of the run in case of fed batch culture, while in case of continuous culture, the product is being taken out at the same time as the media is being added. So this process may be repeated, so that is called repeated fed batch a number of times if the cells are fully viable and productive. So it means that the rest of the culture which is remained within the fermentation tank, so that is being taken up for the next run of the fed batch culture. So there are one or more feed streams but no effluent during the course of operation. So it means that you are not taking out the product during the course of the technique. By manipulating the feed rates, the concentration of the limiting nutrients in the culture can be manipulated either to remain at constant level or to follow a predetermined optimal profile. So it means that when you are adding the media, so you can assess the production as well at the end of the run, and at what stages you are adding the media, like after couple of hours or after 6 hours. So in that case, you can identify that which nutrients are solely responsible for the maximum production of the product, and as a result, when you can identify those nutrients, those limiting nutrients which are very crucial for the production of the product, so then you can add those limiting nutrients at a constant level, like continuously, so that the maximum concentration of those nutrients is always present within the fermentation tank, so that the maximum product can be taken from the fed batch culture. So in case of fed batch culture, the culture volume reaches the maximum. That is obvious because you are adding the media at a continuous rate, so the culture volume will reach to the maximum. Finally, a batch mode of operation is used to attain the final result. So at the final stage, a batch mode of operation means that when you stop the fed batch process, and then you take out the product or from the effluent pipes from the fermentation tank, and then you can identify the final results, that how much product you have obtained. So that was all about this lecture. So I hope that you will understand this lecture, and if you have any problem or if you have any questions, so you can ask me on WhatsApp, or you can give me your feedback through WhatsApp, and I will upload the PowerPoint presentation of this lecture on the KCMS platform, and you can download it from there as well. So yeah, good luck and have a nice day.